Hi, thank you so much for the opportunity to speak here today. That was a wonderful panel discussion, and I'm excited to be part of this larger discussion of how we can bring data-driven science to make meaningful impact in the world and in the space of biomedicine. I'm going to give a little bit of a different talk in the sense that I'm just going to take a moment and always, because nobody wants to be that guy, I'll be that guy. I'll always talk about biosecurity risks, the threats that are looming around us, other things we can think about. I know many people want to put COVID-19 behind us, but I'm just going to take you back there for a moment. So my talk is really going to be about how we can bring genomics and data sciences into thinking about biosecurity risks going forward, and these sort of three pillars we always talk about of detect, connect, and empower. So, first and foremost, one of the things that's unbelievable about where we are today as a society, given how much we know about the world, how much we know about each other, how much data is available, how little we know about the infectious diseases that are circulating around us. Ultimately, if you think about the pandemics that have happened in the last few decades that have really destabilized the global ecosystem, Zika, all of these pathogens have been around for decades. They were not surprises to us. If you think about it, Zika was first discovered in 1947, Ebola in 1976, H5N1 in 1975. Even SARS was originally discovered in 2002. So all of these pathogens, before that big blowout in 2020 and the pandemic that really kind of stopped the world for a bit. But so there's really three dates you can think about. It's the date there was a major pandemic that destabilized economies and caused massive devastation. The date decades before where it was actually discovered, where we had a head start on the thing, but didn't really do enough about it to stop a pandemic or an outbreak from happening. And then the date the thing emerged, which from evidence that we've been looking at are centuries, if not millennia. These things are all circulating in our clinics today, and we're just letting these viruses have a pass. A lot of times doctors tell me the reason that why would we test for these things, we don't have treatments for them. And I always turn that on its head. How could we have a treatment for something we never test for? If we don't have any RSV patients or POOH patients or any of these types of patients to study, we can't develop the diseases. Once we started testing for COVID-19, we suddenly had a whole series of ways that we could think about the diseases. We began to understand long COVID. And so we really need to be in a different place. And ultimately, we know the consequences are very devastating. With 775 million cases that we know of and 7 million deaths with COVID-19, the stakes are high. And outbreaks will really remain part of our present. In 2022, Mpox emerged. 2023, there was an RSV surge. 2024, we have a now H5N1 Mpox cluster and many other things that are circulating. And they'll also be part of our future as we have global, we have a refugee crisis on our hands that we've never seen before. We have destabilization of communities. We have more and more pushes into the environment. We're going to see more of these outbreaks emerge again and again. But we also have the threat of biological weapons and engineered with AI. So in this conference where we're talking a lot about AI, I'll start to just touch upon what we need to worry about in the context of these new genomic engineering technologies and AI. I'm going to actually take a moment and take us back to a study that was done in 2001. So here, what you're seeing here is in 2001, the very same month that the Human Genome Project was published, February 2001. We put out this incredible book that showed every single gene in the human genome. Another group in Australia was discovering something else about what we could do with that kind of information. What they did is they were trying to deal with overpopulation of mouse and rabbit populations in Australia. And so they thought that they could cause sterility in these populations by taking mousepox virus and placing into it interleukin-4, thinking this would cause sterility. Interleukin-4 is something that signals to the immune system to go look for a bacteria, and suddenly it's placed inside a virus, like a decoy. And what ended up happening is they had a group of 3 groups that they were looking at: susceptible mice, resistant mice that were genetically resistant to the virus, and then genetically resistant mice that had already been infected with that same mousepox virus and that had been also immunized. And what ended up happening is mass dissemination. All 10 of the susceptible mice died, all 10 of the resistant mice died, and 8 of the vaccinated and resistant mice died. So what we're seeing here is that it's very possible to use this information to create viruses that will override all forms of our immune systems. And you can imagine this was done 20 years ago. We have far better means of engineering viruses, far more information about the multitude of human genes that could be put together to create a circuit that would kill all mice and override any possible vaccine that we might be able to create. And I can keep going. I said that I'll leave a little time for Q&A because I have lots of thoughts of other things, how AI can really jumpstart all of this. So how will we stop the next pandemic? Well, we have to get much faster. So we in our group talk about a 3-2-1 sort of framework. We need to get to 3 days to detection. 3 days to detection of any novel virus coming into our clinics, coming into our communities. 2 weeks to get diagnostics rolled out all over the world. 1 month to foundational information for countermeasures. Because outbreaks happen quickly. If you give them just a moment to get ahead of it, it becomes an escalating exponential problem. And these technologies need to be accessible in resource-limited situations, just anywhere in the world. So what I'm saying is 3 days to clinically presenting an infectious disease to identifying the pathogen. So we think about this in the context of the Sentinel framework in my own group, this detect, connect, empower, that we need to have diagnostic and detection technologies that work, point of need in your homes for pathogens that we're looking out for, in the hospital to be able to do a broad differential diagnosis, and in genome centers to be able to sequence out and read out, both identify new pathogens and characterize them. We need to have technologies from iPhones in individual homes all the way through to dashboards in our public health to connect that information, and we need to find ways of empowering every actor in the system. And we've been developing technologies across the pace. So I'll just sort of, across the pace in that, I'll just take a moment and talk a little bit more about also what we've been doing for Empower. As part of our work, we co-launched the African Center for Excellence of Genomics of Infectious Disease. We've trained over 2,000 individuals now, actually, from 53 of the 54 African countries. We built a genome center in Edo State in Nigeria. I welcome anyone to go there. We just had Francis Collins and Eric Lander visit, and they were blown away. It's a genome center the likes you've never seen with some of the most sophisticated technologies, 60 apartments if you want to visit there, so please come. But we're really trying to build this community out. Detecting a new pathogen in 2 weeks, the ability to sort of validate and scalable diagnostics. Here, this is just a picture of our center at the Broad, where we created a testing center that ran 37 million COVID tests and supported a lot of the New England region. We need to be able to have these move quickly. Let me just watch the time. So we ourselves in my group have been focused on CRISPR-based technologies, and the reason we like that is because they're incredibly sensitive, incredibly specific, and can be rapidly adapted. So we've been developing point-of-need technology as well as massively multiplex technology based on this. But also, by creating a multiplex platform, we've been able to train models, machine learning models, we run thousands of assays. We've been able to understand how CRISPR guides work and to introduce CRISPR guides not just for Cas13, but for other enzymes, and to create models. And we're also using generative AI, finding that it actually not only improves the diagnostics, but predicts how the virus is going to change and is adaptable to that. We also have been developing technologies to uncover and prioritize cases in real time and to share that information in visualization tools. So this is a multimedia platform called Lookout that we've been developing with our team in Africa, but also been using with the US CDC and the Mass Department of Health, as well as a number of other entities. And ultimately, this is that ability to take all that information and to use it. Everywhere in medicine, we're not using data enough, but in infectious disease, there's another layer we're missing, which is the network, which is incredibly important and why you need this information gathered. And finally, just 1 month from uncovering an outbreak to generating data for vaccines and therapies. And what we need for that is really a focus on platform technologies and the ability to use this information. So I've always been passionate about genomics because I fundamentally say genomics underlies all of the countermeasures that we have for infectious disease, right? It's either the diagnostics target the genome sequence itself or the proteins that are created by that genome sequence, and the same thing with our antibodies and vaccine technologies are fundamentally as the genome changes, these countermeasures are also going to change. So, I believe firmly in the power of genomics to give us that information, to be able to be a blueprint for these pathogens and develop the countermeasures they need. But we also need to be creative in new ways and thinking of new types of platform technologies. So some of the ones that we've been working on in my group are creating CRISPR-based antiviral therapies, knowing that we're going to actually have to have some platform technologies that don't rely on our own immune systems, that would be able to work if some sort of a virus came in and overrode our vaccines, is can we develop treatments that can just work on their own? But we've also been doing a lot of single cell sequencing, really reading out host and viral expression of single cells to really understand how pathophysiologically these things are affecting us and what kinds of countermeasures we could use to improve outcomes. And finally, a lot of work that we've been doing, we've developed a pan-viral approach, a massively parallel synthetic system to be able to read out all of the open reading frames, figure out all of the things that a genome of a virus is creating, find out all the micropeptides that are being created, and then find out which of these are being presented to our immune systems. And we recently had published, or a few years ago published work on that in SARS-CoV-2, where we discovered new novel open reading frames that were presenting to the human immune system and that were eliciting stronger T-cell responses than known antigens. And BioNTech updated their vaccine with that in mind, because some of that missing long-term immunity could be coming from the fact that we're not looking at all of these other elements. So that's fundamentally a quick view of the kinds of ways we're thinking about bringing data science. This is one of the pictures of one of our trainings at the Broad Institute, but this is a very deep collaborative effort that we believe requires collaboration around the world. Thank you.